Hi, everyone. My name is Donnie Day, and I am a PhD student at the University of Toledo. And today I'm going to be giving you a project update on the Exchange Campaign 2 project, Microbial Biomass Collapse. But first, I'd like to give a big thank you to all of our soil senders and their field crews, as this project couldn't have been done without you. I know it was a lot of work going out there to your field sites, collecting soils and sending them back, so I really do appreciate all the hard work and dedication here. Now, we all are familiar with global climate change, and climate change is intensifying precipitation patterns all around the globe. And here in the continental United States, we've seen around a 10 to 20% increase in total annual precipitation since the early 1900s, especially in the Great Lakes and Mid-Atlantic regions. Now, this increased precipitation is predicted to intensify flooding in coastal regions. However, it's still unclear how the duration of flooding will influence microbially driven sea dynamics, as both biotic and abiotic properties vary greatly across different soil ecosystems. Now, in a previous experiment I conducted here in the Great Lakes, where we were trying to isolate the mechanisms associated with dissolved organic carbon mineralization and microbially driven enzyme production, we hypothesized when soils are flooded, dissolved organic carbon mineralization would exceed enzyme production, resulting in sea starvation and subsequent microbial turnover. And this was regardless of topographic position, but microbial turnover timing would be dependent on the availability of soluble C. Now, to do this experiment, we collected soils from a transect that consisted of wetland, transition, and upland. We brought those soils back to the lab, stuck them in microcosm jars, and we subjected them to two treatments. The first being field moist, and then the second being flooded. And then we destructively harvested those soils on days 0, 7, 21, and 55. Now, to test this hypothesis, we measured soil respiration, microbial biomass. We also looked at extractable forms of carbon, nitrogen, and phosphorus, as well as looked at the corresponding C, N, and P acquiring enzymes. Now here I'm showing you a figure of dissolved organic carbon on the y-axis and our harvest days of 0, 7, 21, and 55 on the x, with our two treatments of control and flooded, with the control being in dark blue and the flooded being in this teal color. And we predicted flooding would cause dissolved organic carbon to decrease over time. But instead, we observed an episodic increase that corresponded with microbial collapse. So here I'm showing you the microbial biomass carbon figure, where we have microbial C on our y-axis, harvest days on the x. But I also included our transect here of wetland, transition, and upland. And what you can see is that patterns in microbial C suggest collapse caused increased dissolved organic carbon availability in our flooded treatments. So just to reiterate, on day 21 in our flooded treatments, we saw this increase in dissolved organic carbon that corresponded to a decrease in microbial C. And what was really interesting was this occurred across our transect universally. But C did eventually come down in those flooded treatments. So what happened to this increased dissolved organic carbon availability, and what does this mean for soil C? Well, here I'm showing you a figure explaining the microbial metabolic quotient on the y-axis, harvest days on the x. Once again, our transect is there of wetland, transition, and upland. And our microbial metabolic quotient is the ratio of the amount of carbon dioxide respired by microbes per unit of microbial biomass carbon per hour. And what this MMQ is showing is that we see this episodic increase to respiration in response to flooding across all three zones. So what does this interpretation mean? Well, a high MMQ ratio suggests that microorganisms are respiring at a faster rate than they are incorporating carbon into their biomass. And this is indicating that carbon use efficiency is low. And carbon use efficiency is the ratio of carbon gained to carbon assimilated over a period of time. And so what we're speculating here is that flooding causes microbes to die and turnover. And this turnover releases an abundance of dissolved organic carbon into the environment. But instead of being sequestered, that carbon is being quickly assimilated and processed back out to the environment, or back out to the atmosphere, I should say. So this could have long-term implications for not only sea dynamics, but the overall sea source and sink dynamics. So this raises the question, what is causing this microbial biomass to collapse? Well, we know flooding does influence redox cycles, so do microbes collapse as they step down the redox ladder? Now, here's a figure showing the relative concentration of terminal electron acceptors, commonly used in aerobic and anaerobic metabolisms, where we have oxygen reduction, nitrate reduction, iron reduction, and methanogenesis over time. And what we're thinking is that this could be a result of metabolic inhibition from anaerobiosis. This could be microbe specific, in which, say, denitrifiers are collapsing, or these facilitative anaerobes are collapsing. Or this could just be a driver and change in nutrient availability and or sea starvation. So, getting into Exchange Campaign 2 microbial biomass collapse experiment, we're really asking the question, can we disentangle oxygen limitations, resource availability, and redox effects on availability of dissolved organic carbon and sea mineralization rates? Because our current problem with the first experiment is that we do not know why this microbial biomass collapse happened, or if this response occurs across different environmental gradients. So our objective here is to explore the reproducibility, but also the mechanistic causes and the degree to which these episodic events occur. So to test these predictions, a microcosm experiment was conducted on soils from 9 exchange sites across 3 soil taxa groups. And we focused on the transition area where these soils would experience some type of hydrological change. Now, we focused on soil taxa groups of entisols and septisols and altisols, as they do vary in soil development, with entisols being less developed and altisols being more developed. We had the same setup as the first experiment, where we had 2 treatments of a control and flooded. And we took measurements of greenhouse gas production, such as respiration, CO2, CH4, N2O. We took pore water. We measured dissolved oxygen continuously to get oxygen consumption rates, looked at enzyme activities, and we did a whole bunch of soil extractions. We added in an extra destructive harvest date to really isolate the terms and parameters of what was going on between day 7 and 21. And what we were really trying to isolate here were the sea and nutrient pools, whether they were microbially derived, aggregate derived, or minerally derived. And this was to hopefully understand the potential controls of the microbial die-off we observed in the first experiment, whether it be oxygen limitation, resource availability, or redox-induced transformations. So we measured soil and microbial pool changes, microbial acquisition, and community member turnover in both treatments of the control and flooded. To do this, we looked at aggregate-bound carbon and nitrogen, microbial-bound carbon and nitrogen, aggregate-bound phosphorus and sulfur, and then loosely sorbed pools of those same elements. We also measured physiological parameters such as pH, oxygen reduction potential, dissolved oxygen, gravimetric water content, volumetric water content, as well as bulk density. And then we looked at carbon, nitrogen, phosphorus, and sulfur acquisition and community member turnover by looking at 5 hydrolytic enzymes. The first being beta-glucosidase, which is looking at the release of glucose from the last step of cellulose breakdown. The second being N-acetylglucosaminidase, which is looking at the release of amino sugars from chitin and other glucosaminopolymers. The third being leucine aminopeptidase, or looking at the release of leucine and amino acids from peptides and proteins. And then we have phosphatase and aryl sulfatase, which is looking at the release of inorganic forms of P and sulfur from organic phosphorus and sulfur. We also wanted to consider the redox effects, as mentioned before, so we measured terminal electron acceptors, and we did this only in our flooded treatments. So pore water was collected for nitrate, ammonium, and phosphate, iron 2 and iron 3, and SO4 and H2S. And this is what we were predicting would happen, and we're hoping to be able to pair this up with the microbial biomass collapse we saw in the first experiment. So project timeline and next steps. So last October, we sent out exchange kits. We received samples in mid-November of 2023. We processed those samples immediately for all of the things we've mentioned before. So we have a pretty extensive soil data set for Exchange Campaign 2. But we didn't actually start the experiment until the end of January 2024. And that experiment went all the way out till June 2024. So we just finished the experiment, which is good news. We've processed all of the greenhouse gas data, dissolved oxygen data, and enzyme data. And we're currently in the process of running samples for the analyte pools of, soluble, aggregate, adsorbed, and microbial. So we're still in that process. We're hoping to finish that up by the end of November and then get in to start, our data analysis, making figures, and then actually writing up this thesis chapter, but also starting the idea of a manuscript to follow. And so we're expecting 2 manuscripts coming out of this. The first is with the overall experiment here with Exchange Campaign 2, microbial biomass, but we're also hoping to probe some additional questions with EC2 data, especially from the intake samples. So this is my call to arms for opportunities for collaboration, as these few tests are not what I'm going to be able to get to, but I do think it would add a lot to the data set. So first, we're looking at texture analysis, because right now we're only able to characterize the soils by their soil taxa groups. So texture analysis would provide sand, silt, and clay ratios, and this would allow us to better characterize soils beyond that taxa group. Second, looking at CHN analysis. This would provide total organic and inorganic pools not captured in the current extractions. And then third, which would be pretty exciting if we get the data we're predicting, is to go in and look at FTCIR and NMR analysis. And this would allow us to provide some chemical profiles of both organic and inorganic compounds over time. Now, with that being said, those were kind of my top 3, which I think would add a lot to this data set. However, I'm open to input and collaboration for the final manuscript. So if anything I've said today was exciting, or you think if you have any questions or concerns, please reach out, as I would be excited to share, what I have, and if there's something that you can provide to the project, please reach out. Then lastly, I mentioned probing additional questions with Exchange Campaign 2 soil samples, as sadly not all the soils made it into the experiment, and those soils being the histosols, but we did collect a lot of data, especially on the intake. So if you have an idea or a research question using this data set, reach out. My email is donnie.day@rockets.utoledo.edu, or please contact coastal.exchange@pnnl.gov. With that, I want to thank you for your time. I really appreciate the opportunity to contribute to the larger Exchange consortium. So until next time, when I have some data to talk about, I'll see you all later.